Hello everyone, let's continue from where we left in the last class. We were discussing about two-component system and taking reference of cytokinin signaling with respect to plant development. Let's look further and deeper into it and see what all ways plants respond using two-component system. If you recollect some of the things which we discussed in the previous class, the idea was that normally plants use hybrid two-component system, if I say TCS. You also notice that there are variants of two-component system in the sense that they were pseudo kinase and pseudo response regulator. We also saw that cytokinin, the hormone which is important for plant development, it affects root and shoot development depending on what concentrations are present, where it is localized, and what is the relative concentration of oxygen over there. As well as we will discuss how ethylene responses are mediated by two-component signaling in today's class. Remember that we will need the histidine kinase, response regulator, and since it is a hybrid type of two-component system, we will need histidine phosphotransperases. Also remember, this is a hybrid type of two-component system, so there will be a phosphor relay. And the variants which will be there, they will impart next level of complexity in the signaling. So we also discussed in the previous class that there are multiple different plants which have been studied, whether multiple different plants have been studied, whether it is Arabidopsis, the model dicot, or rice, or soybean, or corn. Multiple different of them have been studied to analyze whether the components of two-component system present in all of them, what is the relative abundance, and how many of these molecules are there. So what you will notice that more or less every plant species which have been studied in this context has different components, whether it is the histidine kinase or there are variants of histidine kinase. Remember the variants of the histidine kinase, where the histidine residue which accepts the first phosphate because of hydrolysis of ATP, that is missing, and that makes it a complex system. Then we also notice that there are different types of response regulator. So the analysis of response regulator coding genes in all these different species suggests that they are abundantly present. Whether it is type A, remember these were the small response regulator, minimal domain containing, having one aspartate residue where phosphate can be transferred, or type B, which were little bigger by virtue of having another domain which will catalyze different type of reaction. So these numbers which you see here are reflecting the genes which are coding for those components. There is additional type of response regulator which is present in these plants, is that type C. We did not discuss that much more in detail because lot of studies which we will encounter further, we will see type A and type B are doing most of the job. As well as there are response regulator variants. If you recollect, the response regulator variant, which are basically the aspartate residue in the receiver domain, is missing or mutated or replaced with some other amino acid. Now people have studied here, I am showing you the example of two different plant species. One is Arabidopsis, where different chromosome, we saw different genes in the previous slide, that different genes for histidine kinase or response regulator. What has not been shown here is the histidine phosphotransphorases that also have been mapped on to each of these plant species, that how many genes are coding for response regulator, histidine phosphotransferase and histidine kinases. When people have analyzed the distribution of these genes over different chromosomes, so we know that Arabidopsis has 5 different chromosomes, and the numbers here on the left, this is chromosome 1, chromosome 2, chromosome 3, 4 and 5. You can see that almost equal distribution seems to be there on each of the chromosome. In case of bacteria, when we saw 2-component system, response regulator and the sensor kinase, in many cases those are regulated by operon, and single operon is coding for the components of the 2-component system. But here what we notice that the distribution, the operon concept does not apply in plants. So here we have more of equal distribution of these genes on different chromosomes. Similarly, if you look at the tomato, there are 12 chromosomes and there is distribution of these genes which are coding for sensor kinase or response regulator or histidine phosphotransferase on all these chromosomes. So what it suggests that the 2-component system is working prominently in plants. There are multiple genes which code for the components of TCS, and these components are evenly distributed over different chromosomes present in the plant species. Now you can see that what exactly it means, that if you have consider a situation of chromosomes like this being the centromeric position. Also, not only that the genes are distributed evenly on different chromosomes, but they are also evenly distributed on different arms of the chromosome, whether it is P arm or Q arm. It is not very evident here, but if you make something like this as a P arm and Q arm, the genes are located all over it. There is no bias towards one arm or towards one chromosome when the gene distribution for components of 2-component system comes into effect in various plant species. Since Arabidopsis, a model dicot plant, has been studied in great details, we will find more references with respect to Arabidopsis when we discuss more of the signaling by TCS, particularly with respect to cytokinin or ethylene. So let's see how a simple 2-component system is working in plant, what are different components which are there and how they mediate the signaling. We will see first a model that how it will work, and then we will take specific example how it moves forward. So think about this scenario where you have this plasma membrane on which there are 2 different types of receptors. If I say receptor type 1 and this I say receptor type 2, they are present. If we go back little bit and if I ask you to recollect that one of the event which I mentioned in the signaling to initiate that and take the signaling further down inside the cell was the binding of the ligand to the receptor and then dimerization. So what is shown here are 2 different types of receptor probably responding to the same signal, and the signal here you can consider as cytokinin. Now in this situation as shown here is Arabidopsis histidine kinase 2 or 3. They will dimerize. Also remember this domain which was internally present where histidine residue is found, it is also called DHP, dimerization and histidine phosphorylation domain. So what will happen is that once the cytokinin binds to this class of receptors or to this class of receptors. They will dimerize, and that dimerization induces the kinase activity. What will kinase do? It will autophosphorylate itself as the first measure. And since these are hybrid type of histidine kinases, they will transfer the phosphate to the aspartate residue. Since this phosphate has been transferred to the aspartate residue, it will look for a molecule which has the histidine residue, and that's where you will have histidine phosphotransperases. Remember one more thing which I told earlier, that the transfer of phosphate group may lead to change in its conformation and change it or switch it from inactive to active state. So you can consider that this Arabidopsis histidine phosphotransferase, which was inactive, when it receives the phosphate group from either of these receptors which are activated, it will be in this situation and active. Now this molecule will move into the nucleus. In previous class we saw different domains, different motifs which are present. For things to move inside the nucleus, you need nuclear localization signal. So if upon phosphorylation the histidine phosphotransferase moves inside the nucleus, it will have the NLS type sequences. But the signaling does not finish here by virtue of the histidine phosphotransferase moving into the nucleus, because you remember that signaling moves from histidine to aspartate, aspartate to histidine, and the final response regulator or master regulator must receive this phosphate to culminate the signaling process. So your histidine phosphotransferase has moved inside, and then it will look for something which is present over there. What is exactly present over there is this master regulator. This master regulator, as the domain arrangement suggests, that it has the DNA binding domain and an activation domain. Where are these domains utilized, or where are these domain presence? Where are these domains present? They are part of transcription factor. So we are taking one example here, that how cytokinin signaling is mediating responses. So what we notice, that this phosphorylated histidine phosphotransferase has moved inside, and it will be looking for the response regulator where conserved aspartate residue will be there. And that conserved aspartate residue is present in this master response regulator, which are in the family of authentic or Arabidopsis response regulator 1, 2 and 10. Now this molecule is already present inside the nucleus, and it has to be stopped from its functioning. So a repressor binds to it. These are different types of regulation where we studied in previous classes, that how signal will be regulated, how your effector will be regulated, how your receptor will be regulated. So if this transcription factor, which has the ability to bind to DNA, is already present inside the nucleus, its activity must be repressed so that this gets turned on only in the presence of right signal, and that is being achieved by the binding of the repressor. Once this phosphorylated histidine phosphotransferase moves inside the nucleus, it transfers the phosphate group onto this aspartate residue. Because of that, this repressor is released. Remember that these modifications play a subtle role in switching the effector molecule from its inactive state to the active state. The inactivity here was because of the binding of the repressor. Phosphotransfer event, which happened here, releases the repressor, and now this transcription factor is active. What this transcription factor does, it actually takes it and binds to the response element regions, which allows the transcription to happen. Transcription of what? The response regulators. This is like feedback mechanism. The signaling started with the cytokinin, and it terminated into the transcription of response regulator, this will be sensed as the culmination of the signaling. That's why the molecules which are produced, they will inhibit the step of transcription as well as they will inhibit the phosphorylated state of this histidine phosphotransferase. So you can imagine that as soon as histidine phosphotransferase transfers this phosphate residue onto the aspartate residue, the dephosphorylated version AHP12, which is dephosphorylated, will now move out of the nucleus. So nuclear localization signal which was there, that is sensitive to the phosphorylated form of the AHP, and that's where the shuttling between nucleus and cytoplasm is regulated. Now let's look at what exactly this eventually will do. So we saw you got the type B response regulators synthesized. If I go back, this is your type B response regulators. Now what they will do, they will engage in various activity. The activities which will ensure that shoot and root development is regulated. So the transcription which happened, this will affect the shoot and root development. Sometimes there are molecules which directly, without engaging the histidine kinase signaling, called cytokinin response factors, they move inside the nucleus and help in this entire process. So integration of the signaling happens. We remember, if you remember, we discussed that also, that how different signaling can be integrated to mount the best response. So one of the things which is prime to the function of cytokinin is root and shoot development, that is regulated by the synthesis of these type B response regulators. Further, they actually work through type A response regulator. So when before I come to type A response regulator, I will just elaborate gently what happens here in the root and shoot development, that the type B response regulators which are generated, they will probably allow the more transcription of desired genes, which will promote the shoot development in right set of tissue. In root development, the tissues which will be a different type, it will be a different response regulator governing the transcription of next molecule. As well as what will happen, that it will affect the seed development, as well as the vascular bundle differentiation. These are known functions of cytokinin. What we are noticing is that by virtue of activation of two component system, different pathways are set into motion which will ensure that different aspects of plant development, whether it is root, whether it is shoot, whether it is seed or vascular bundle differentiation, all are working together. One thing which I started telling you about the type A response regulator, these are generated and they are shuttled into another response which we call as photomorphogenic response. So what will happen over there, that these type A response regulator will engage into the signaling which is mediated by phytochromes to generate the photomorphogenic response. We will see more examples of that in the coming slide, but one thing which cytokinin ends up affecting is the meristem properties. So all these type B response regulators which are being generated in response to cytokinin binding to its receptor on cell surface, you will see these processes getting activated. Now you may be confused that how come a cell which has this cytokinin responding histidine kinase is going to do all these processes. Remember I said that there are multiple of these kinases which are present. There will be cell specific or tissue specific histidine kinase which will be responding to the cytokinin, and it will have one of the programs which will be regulated over there. So if it is a shoot cell, it will be regulating the shoot development. If it is a root cell, it will be regulating the root development. If you are talking about the meristematic tissue region, it will be affecting the meristem formation and so on. So you can relate that how single type of sensor kinase or histidine kinase is responding to the same molecule and giving different outputs. So now if we look at the two component system and the phytochrome signaling, if you recollect, phytochrome was a variant type of histidine kinase receptor, which means that it might have that typical histidine residue mutated to something else, arginine or lysine, or we don't know, there could be many of these options, right. Also phytochromes help in the process of photomorphogenesis, which means that they respond to light. Here the protein phytochrome exists in two forms. If I say phi and take example particularly of phytochrome B, so depending on which light is present, it is in active state or in an inactive state. So if you have red light falling on the phytochrome B, it switches into the active mode, and when you have far red light, it switches back into the inactive mode. We can understand that in dark there will be far red light, and that's where it will be in the inactive mode. So let's see how cytokinin dependent signaling is coupling with the phytochrome signaling. We already noticed that cytokinin receptor, upon binding to the cytokinin, they will dimerize, phosphorylate on histidine residue and aspartate residue. This will allow the AHP to be phosphorylated. AHP moves into the nucleus where it transcribes the response regulators. And we know that these response regulators are in two types, B type and A type. So these A type response regulators, they actually engage in the crosstalk with the phytochrome signaling. What they do is that they help in interconversion of phytochrome into different forms. If I say phi B, if I just label it as A and I as active and inactive, so in far red light this is converted into inactive form, and in red light it is converted into the active form. So this A type response regulators, which are generated as a consequence of cytokinin signaling, they work through this phytochrome B form which is active, and then induce it to its inactive state. So the reversal which is happening here is, if I just redraw this portion, so what you will notice is that so from active to inactive state is basically what is being induced by the far red light, and inactive to active state is being induced by the red light. So this conversion of active state to inactive state is inhibited by the A type of response regulator. So you can say that as long as A type response regulators are synthesized, they will inhibit the conversion of phytochrome B into its inactive form, thus they will support the photomorphogenic responses, which means that this A type inhibits this process. What it means that you have more time available to the active form of this phytochrome B. So what you notice is that signaling which started with cytokinin binding to its receptor on cell surface culminated into the transcription of different types of response regulator, some being B type, some being A type. B type engages into the regular responses which are required by the plant, which is a consequence of cytokinin direct signaling. The type A response regulator, they, apart from working what they are supposed to do, they impinge or they connect it to the phytochrome signaling, where they inhibit the conversion of active form of phytochrome B to inactive form of phytochrome B, which gives more time to the active form of phytochrome B and thus mediate the phytochrome mediated signaling. We will see more of that in the next class. And what we have understood in this class is that plant species, different types, they utilize the two component system, they have abundance of it. Not only that, every component is present in the plants, but also the variants are present in the plant, whether it is histidine kinase variant or response regulator variant. The two component system, which is hybrid type utilized by cytokinin, they mediate multiple responses, whether it is shoot development, root development, vascular differentiation, or meristem maintenance. We will see how these are connected to another type of signaling which plants exhibit normally, and the cytokinin response further induces a crosstalk with photomorphogenic response through the type A response regulator. Thank you.